In order to persistently move in the same direction for extended periods, migrating fibroblasts must remain polarized with a protrusive front and a retracting rear. PI3K's and its phospholipid products are polarized toward the front of the cell, but their precise function in persistent fibroblast migration remains unclear, as Jason Haugh from North Carolina State University in Raleigh explains.
We had seen in previous work that PI3K's signaling was very dynamic. It seemed to be very strongly correlated with membrane protrusion, but it had a certain time scale associated with it that couldn't explain the directional persistence of the cells. In other words, the hot spots of PI3K's signaling had a time scale of a few minutes and move around to different locations in the cell, whereas the directional persistence would be an hour or longer. So it wasn't clear how we could connect those two time scales.
To better understand how rapid changes in PI3K's signaling and membrane protrusions help orient cell migration over longer periods, Haugh and colleagues, led by postdoc Eric Welf, used turf microscopy to monitor signaling and membrane dynamics in fibroblasts expressing GFP-tagged AKT-PH, a biosensor that binds to the products of PI3K's.
So it wasn't clear just from watching a movie how the dynamics of these different PI3K's hotspots were linked in space and time. And by making a spatiotemporal map, we can look at protrusion and retraction of different regions of the cell with respect to time, alongside the dynamics of PI3K's signaling.
Both PI3K's signaling and membrane protrusion and retraction showed characteristic tracks on the spatiotemporal maps that followed the fibroblasts' long-term movements. The tracks revealed that migrating fibroblasts make large turns by branching off a protrusion at their leading edge and pivoting to follow that branch in a new direction. Because PI3K's activity localized to these branching protrusions, Haugh and colleagues thought that the kinase might be involved in cell reorientation. They therefore blocked PI3K's signaling using either an inhibitor or a dominant negative version of the kinase's regulatory subunit P85-alpha.
In either case, the cells became more elongated and failed to show these successful branching events. They were still producing branched structures, but those branched structures were unstable. So very rarely did they result in reorientation of the cell.
PI3K's inhibition prevented fibroblasts from making large turns without affecting cell speed or the frequency of forming branching protrusions. Dual imaging of PI3K's activity and F-actin revealed that actin polymerization overlapped with signaling hotspots. Protrusions still formed when PI3K's activity was blocked, but they failed to broaden and stabilize, perhaps because PI3K's signaling helps activate the ARP2/3 complex to promote the formation of a branched actin network. Unable to stabilize, the protrusions eventually retracted, without changing the cell's orientation.
By looking at the spatiotemporal maps in more detail, we were able to tease out that the PI3K's signaling accumulates a minute or two after the onset of protrusion.
The researchers then used a photoactivatable version of the RAC-GTPase that induces local membrane protrusions in response to blue-green light.
This was a way to corroborate our correlative findings between protrusion and signaling. We wanted to induce protrusion using the photoactivatable RAC and see if PI3K's would be localized in response. And indeed that was the case. 95% of the cells showed this strong localization of PI3K's signaling in response to photoactivatable RAC.
Like branching protrusions at the cell front, PI3K's wasn't required for the formation of RAC-induced protrusions. And kinase activity didn't relocalize if RAC-induced protrusions were blocked using an inhibitor of actin polymerization, indicating that signaling hotspots are directed by the protrusion rather than by RAC itself. These experiments, all performed in randomly migrating fibroblasts, suggest that PI3K's helps cells turn by stabilizing branched membrane protrusions at the leading edge. but can PI3 kinase-mediated branching and pivoting help fibroblasts reorient in response to a chemoattractant gradient?
We looked at cells that were migrating in response to PDGF gradients that were presented through the loading of alginate microspheres with PDGF. And then we watched the reorientation of the cells relative to the orientation of the PDGF gradient.
Under these conditions, fibroblasts, such as this one, often formed a branch protrusion at their leading edge and maintained strong PI3 kinase activity in the branch aligned with the PDGF gradient, probably because the